In this lecture, we are going to discuss human herpes virus 8, or you can say Kaposi sarcoma associated herpes virus, because it's an important agent of the Kaposi sarcoma. So let's get started. First of all, we will look at the structures of the human herpes virus 8. So basically, it is an envelope virus. Outside, you will see the presence of an envelope. Okay, and inside the envelope you will see the presence of the capsid that is actually made up of protein, and the shape of the capsid is icosahedral, means 20-sided, and inside the capsid you will see the presence of the double stranded DNA molecule. Okay, next if we look at the types of the human herpes virus 8, so you will see six most important subtype: type A, type B, type C, type D, type E, and type F. So these are basically the six most important types of the human herpes virus 8. If we look at the transmission, how human herpes virus 8 transmit from one person to another, so it actually transmits through the saliva. It can be transmitted through the sexual contact and due to the transplantations of the organ. So during the transplantations, the human herpes virus 8 can be transmitted from the one person to another. Okay. So what type of cells mostly affected by the human herpes virus 8? You will see the two most important cells, B lymphocytes and the endothelial cells. These are the most important cells mostly affected by the human herpes virus 8. If we look at the replication, so you will see the two most important cycles of the virus, lytic cycle and the latent cycle. In the lytic cycle, you will see the virus will first of all affect the cell, and as a result, you will see the productions of the different replica of the virus. Okay, you will see the lot of productions of the virus happen inside the cell as a result of lytic cycle. But in latent cycle, you will see the virus will affect the cell, but the genome of the virus will remain inside the nucleus of the cell as episome. But the latent cycle is more dangerous as compared to the lytic cycle, because in the latent cycle you will see the cell can convert into the malignancy. So that is the important thing. Okay, so you will see the three most important process that actually happens in the latent cycle, and as a result the normal cell converts into the malignant cell. First of all, the viral gene 1 will produce the LANA, latent associated nuclear antigen. Okay, so this antigen ultimately inhibits the productions of the p53 tumor suppressor protein. Okay, so inside the cell you will see the presence of the p53 tumor suppressor gene. Gene will produce the messenger RNA, and after the messenger RNA you will see the productions of the protein. Okay, but LANA, latent associated nuclear antigen, that is actually produced by the viral genome, this LANA will block this process, means transcription, because formation of the messenger RNA from the gene is called transcription, and from the messenger RNA you will see the productions of the protein, and that process is actually called the translation. So LANA will inhibit the transcription of the p53 tumor suppressor gene. So as a result you will see no productions of the p53 tumor suppressor protein, and ultimately the cell go towards the malignancy. Okay, next, viral gene 2 will produce the cyclin D. Cyclin D basically overrides the control of the cell cycle growth, and as a result you will see growth of the cell. Okay, next, viral gene 3 will produce the protein that actually block the FAS death cycle of the cell. After the inhibition of this cycle, the cell will survive, and ultimately the infected cell will grow. So here are basically the three mechanisms through which the cell starts growing and ultimately go towards the malignancy. And these three processes will happen specifically in the latent cycle, not in the lytic cycle. Okay. Next, if we look at the pathogenesis. Okay. In the Kaposi sarcoma, KS, you will see the formation of the lesions on the skin. Okay. So, if we take the biopsy of the skin and look in microscope, we will see the spindle shaped cells. Okay, spindle shaped cell. But the origin of these spindle shaped cell in the biopsy will be endothelial in origin. Means that these spindle cells in the lesions of the Kaposi sarcoma, you will see the origin of these spindle cells will be endothelial, or it can also be lymphatic as well, vascular endothelial cells, or it can be lymphatic origin. And along with that, you will also see the presence of the red blood cell as well in the biopsy. So that is the important thing about the Kaposi sarcoma, the most important disease caused by the human herpes virus 8. Okay. And next, if I say this is the infected cell, virus infected cell. Okay. So inside the cell, you will see the production of the two most important protein, MIR1 and MIR2. Modulator of immune recognition 1, modulator of immune recognition 2. These are the proteins that will suppress the expression of the major histocompatibility complex 1 on the surface of the cell, because major histocompatibility complex basically present the viral peptide on the surface of the cell, and as a result cytotoxic T cell, or you can say CD8 positive, or you can say cytotoxic T cell, will destroy the whole virus infected cell. But due to the productions of the MIR1 and MIR2 by the virus, you will see the suppression of this major histocompatibility complex. And as a result, due to the suppression of the major histocompatibility complex, there is no representation of the viral antigen on the cell surface. And ultimately, this virus infected cell will survive in our body. Next is the manifestation. So as we have discussed, the most important disease caused by the human herpes virus 8 is the Kaposi sarcoma, but it can also cause other diseases like multicentric Castleman disease, primary fugal lymphoma, inflammatory cytokine syndrome. These two, multimeric and the primary fugal, is actually related to the B lymphocytes. Okay, as we have discussed, this virus affects mostly B lymphocytes and the endothelial cells. If we look at the lesions, what type of lesions you will see in the Kaposi sarcoma? You will see the reddish to dark purple lesion. Okay, these lesions can be flat to nodular. Basically, you will see the three stages of the lesions. You will see the patch formation, and after that you will see the formations of the plaque, and after the plaque you will see the formations of the nodules on the skin. So it can be produced on the skin, it can be produced on the oral cavity, GIT tract, and the lungs. So basically these are the location you will see the formations of the lesions caused by the human herpes virus 8. You will see four most important type of Kaposi sarcoma. First is the classic, that is mostly seen in old people, and you will see the lesions formations specifically on the legs. Okay, and it is actually slow growing Kaposi sarcoma. Next is the endemic. This Kaposi sarcoma mostly seen in Africans, and it will affect mostly men. Okay, next is the epidemic. You will see mostly HIV people affected by this. That is you will see the Kaposi sarcoma in HIV patients. Okay, next is the immunosuppressive therapy related Kaposi sarcoma. This is mostly seen in transplant patient. So here are basically the four most important type of the Kaposi's sarcoma. Next, if we look at the lab diagnosis, so we can diagnose it through the biopsy. We can take the biopsy of the skin lesions, okay, and we will check it in the laboratory, skin biopsy, and we will detect the spindle cells, okay. Next is the PCR. We can detect the DNA of the virus in the spindle cells because the DNA will be present in the spindle cells of the biopsy. So we can also go towards the PCR, and we can also go towards the immunohistochemistry as well. And next is the serology. We can detect the antibodies in the blood of the patient. And next is the lesion. So lesion will vary according to the patient. For the localized lesion, you will go towards the surgery or intralesional vein blasting. And for the extensive lesions, you can also go towards the radiation therapy. And for the advanced Kaposi sarcoma, you can also go towards the chemotherapy like liposomal doxorubicin and the daunorubicin. And in HIV affected people in early stages, you can also go towards the hard, highly active antiretroviral therapy. So here are basically the different treatment plans, but these treatment plans will be vary according to your physician. So this is all about the human herpes virus 8 or Kaposi's sarcoma. So if you still have any question, you may ask in the comment section. Thank you so much.